Hello, and thank you very much for this opportunity. My name is Maria Emfietzoglou, and I'm a research fellow at Mass Pioneer. In our recent study in Ophthalmology Science, we showed that the inner plexiform layer substrata can be seen with commercial OCT and are affected by aging. Now, we all know that the inner plexiform layer contains the synapses between bipolar cells, ganglion cells, and amacrine cells. But something that isn't commonly known is that these synapses are actually arranged into 5 distinct sublayers. What is amazing is that Cajal, the Nobel Prize winner, over 130 years ago, was able to see this using only primitive tools, and he actually drew these 5 sublayers in 1893. Now, later studies have confirmed this with modern histology, and we now know that this stratification isn't just structural, it is also functional. Now, some experimental OCT systems like visible light OCT have shown these sublayers before. On high-resolution OCT, these sublayers can be seen because they appear as an alternating pattern of hyperreflective and hyporeflective bands. But these experimental systems aren't available in clinics. What we found in our study is that some commercial OCTs can actually show these sublayers too. One of the best was the enhanced HD line scan from Optoview Avanti. Using this scan, we analyzed the IPL from healthy people, and we found a consistent pattern of 3 hyperreflective and 2 hyporeflective bands, which matched the 5 known sublayers. And then we asked what happens as people get older. And we found that aging doesn't just make the IPL thinner, it also attenuates the intensity profile. The hyporeflective bands become more hyperreflective, the contrast between the bands drops, and the 5-layer pattern becomes harder to see. And this makes sense because we know that aging affects the synapses. Now, how did we do all this? To answer that, I will hand over to my colleague, Victor Correa, who is now an ophthalmology resident at the University of Missouri in Kansas. Victor, can you tell us a bit more about the methods we used?
Yes, thank you, Maria, very much. First challenge here was to extract meaningful and reliable data from these scans. The HD scan from the commercially available OCT that we used, we could see that IPL was substratified, especially on the the area between the macula and the optic disc. So we focused on that area. We imported the scans into ImageJ initially. We selected manually the inner plexiform layer, which, by the way, is the only manual step in our whole pipeline. And then we extracted all the pixel values and transformed by Z-score, so we could compare between eyes and with different patients as well. And after we extract all these values, we transform it into the Python pipeline, where we could, enhance the contrast, so we could, better determine all the values on these different layers. One thing also that we could show with that is that not all the patients we could see those 5 sublayers, especially on the older patients, like Maria was saying here before. And the thing is about this other pipeline might sound a bit confusing, where we could first show the first hyperreflective layer, then hyporeflect bands, and so on and so forth. But within the Python pipeline, it should take a couple seconds only, and it's very user-friendly. So I can see in the future this to be adopted into OCTs, so we can give clinicians some metrics to analyze our patients. We could identify a lot of information with that, especially the percentage of patients that we show this subsegmentation, but also what's the value of each sublayer regarding the signal intensity, and most importantly, the contrast between each layer. And with all this information, we could then compare between groups and show that aging would, reduce the contrast between these layers and making it harder to identify them. Also, importantly, we would test for the reproducibility of our study. So we compare different analyses on the same eye and then between eyes, and we find that it was pretty reliable and reproducible. So now go back to Maria here to talk a little bit about the critical importance of these findings.
Yes, so this work is important because the IPL offers a way to observe synapses in the human nervous system non-invasively and in real time. And since synaptic dysfunction often happens before overt neuronal cell loss, changes within the IPL could be a very early sign of neurodegenerative diseases. Things like glaucoma, Alzheimer's disease, Parkinson's disease, or multiple sclerosis. And there are prior studies that have shown that IPL changes in these diseases, but most prior studies have looked at IPL as a single layer, and they have focused on a very coarse metric, IPL thickness. But this may miss earlier changes, as IPL thinning may not be the first sign of pathology. So, what we are proposing here today is something more detailed. It is analyzing at the individual IPL sublayers to get more granular information about these synapses. This could help us detect diseases earlier, track how they progress, and as a group in Dr. Vava's lab, we are already working on that next step, exploring IPL microstructure in neurodegenerative diseases. And in fact, we will be presenting our work about IPL in multiple sclerosis this year in ARVO. Thank you very much for your time.
Thank you very much.